Well, welcome back, everybody. We're into the final session of today. So we have a series of short talks, 6 short talks, first of which is by Paul James. Paul's presentation title is Development of Synthetic Biology Toolkits for Non-Model Organisms. Over to you, Paul.
Thanks very much, Gary. So hi, everyone. Yeah, I'm just going to give you an introduction to some of the work we have done trying to engineer non-model organisms. And hopefully, as you can see, as we go through these sessions with the VCFs, that we have a main aim of the theme, which is to take a building's waste and turn it into something useful or some high-value products. And where my interest sits is in engineering those microorganisms that sit within and hopefully become these highly efficient cell factories that sit within the middle of. So hi, everyone. Yeah, I'm just going to give you an introduction. Sorry, can everyone hear? I've just got myself playing back to me there.
Yeah. 2 Pauls. Unbelievable.
Yeah, I mean, yeah, everybody's worst nightmare. Right, okay, sorry. So I'm interested in the enzymes and the microorganisms that will hopefully be involved in these highly efficient cell factories. So for those of you who don't know, I'm interested in using synthetic biology or engineering biology techniques, and they really are the application of engineering principles to biology. And one of the central pillars of that is this design cycle. So you can see that what happens is we look for the organism that we're interested in. We find a pathway that we're interested in. So in the top left-hand corner here, you can see the an alkane producing pathway that I worked on previously. We pick a microorganism and then we try and build the parts that we're interested in within this microorganism. And then we have a test phase where we're looking for those metabolites, looking to see what's been used up, and then hopefully we can learn something from those experiments, and we try and do that in a statistical manner so that it be applied. And then hopefully we go through so a few iterations of this design cycle, and we end up with a functioning system that can be implemented. So why am I interested in non-model organisms? Well, you know, model organisms are brilliant. They've been well studied. They're usually domesticated for laboratory use. They have a high number of genetic tools that are available. So why do we really want to move away from that? You know, everything's kind of there for us to start with. Well, I suppose, you know, they usually have to be engineered to perform all of the functions of interest. So, you know, in these complex systems, we're going to want to take waste that's usually quite recalcitrant and then turn it into something. So if I've got to engineer it at both ends of that workflow, then you know that's going to make the job a whole lot harder. Whereas non-model organisms are obviously quite different. They're not very well studied in the lab. We have very few, if any, genetic tools sometimes for them. They can be difficult to cultivate in a laboratory environment. So some of them are isolated from field studies, but then when we try and grow them on their own in a laboratory environment, that can be quite difficult. But usually they're highly adapted to the environment that they're found in. So, you know, we are interested in them because they've either been seen to grow on a substrate that we're interested in, or you know, they can produce products of interest. So they're usually already able to do some of that work that we require. And then just down the right-hand side here, I've got some examples. So Vibrio nitrogens is something that people are interested in with synthetic biology. It has an ultra-fast growing time. It can double sort of every 10 minutes. You know, we've got Pseudomonas, which is interesting because of some of the metabolites it produces. It also has high tolerance towards toxic compounds and things like that. And then Lactococcus lactis, that's been known now for producing some flavorings and texturizing compounds. So one of the organisms I'm interested in is Geobacillus. So it's rod-shaped, gram-positive, aerobic or facultative anaerobe, and it's usually found in thermophilic environments. And so it was originally sort of a lot of enzymes for biotechnology were isolated from there because it had a lot of interesting functions, but also interested from the thermophilic standpoint. But you know, this is it's quite a large genus. So the Bacillus stock center has over 168 strains, and there are a lot more available. And they've been isolated from a wide range of different environments. So oil fields, where one enzyme that was added into Geobacillus meant that it was able to grow on oil contamination, you know, right the way through to compost heats and hot springs. And as I said before, you know, it's been known to grow on a wide range of sugars, so that makes it very interesting from that standpoint. So, you know, we were interested in this organism, but we thought, what do we have to do to be able to engineer it? And we were interested in looking at a range of different methods to try and build up a toolkit that we could then engineer it to perform the things that we're interested in. So to start with, we wanted to have a really good grasp of all the genomics. So we wanted to look at across genus, not just at the one species that we were interested in, but all of the genomes that have been characterized and some that we added ourselves. And the idea was that we could start to build up some of these keg maps. You know, we could start to look at the natural metabolisms that are taking place within the organisms. So we could either mine it for interesting enzymes, or we could start to sort of build on these metabolic pathways. And we're interested in things like codon usage. So if we're trying to express genes of interest, you know, are we going to need to look at codon usage as we move through? So we also wanted to be able to get stuff in. So we were interested in sort of origins of replication and selection markers that could be used to develop a suite of vectors that meant we could, you know, we could put more than one vector in, or we could tailor the vector towards the sort of process that we were trying to develop. And we also wanted to look at things like standardized assembly. So we want to be able to put lots and lots of parts together in a combinatorial manner, and really having a cloning system that's designed to do this makes this a lot easier. So one of the main focuses of our research today is focus on trying to characterize some genetic parts so that we can get sort of a suite of promoters that will allow us to have a range of different gene expression levels. So we wanted them to be predictable, and we wanted them to have a range of strengths, because there's no good only being able to turn everything up to 11. But we also want to, we want them to work as the same in all situations. So we started with promoters. What we did was we took 100 base pairs upstream of all coding sequences within the all the genomes that we had available. If they overlapped with any other coding sequences, obviously we got rid of those. We then ran it through some transcription factor prediction sites. You know, these were a bit fuzzy because they were from E. coli, but you know, if it had anything that looked like it could be a transcription factor, they ended up in the bin, and we ended up with around 1,400 sequences. So we collapsed these down to around 21 clades with no more than 150 sequences per clade. And that was just arbitrary, really. That was just to try and get things that had a similar sequence together so that we weren't sampling from things that were too similar. And then we randomly selected around 5 sequences from each clade and then tested those. And as you can see, we did end up with a wide range of different strengths of promoter. But one thing we did see was that there was very little correlation between expressing GFP and expressing M-orange. So what we saw was there very little, the promoters were very context dependent. So it depended highly on the coding sequence that was sat in front of it. So one of the things that Matt Rogan, my PhD student, is now looking at is there a way of getting rid of some of that context dependentness? You know, can we stick an insulator in there that means that the promoter sequence that we're looking at, we can have a predicted strength for it, and it will work at that strength with all sequences. He's also interested in characterizing some other genetic parts. So he's looking at things like the ribosome binding site that came out of those promoter sequences, and looking at seeing if we can engineer the terminators to have that predictable gene expression level. And also he's looking at things like signal peptides because some of the time we might want to obviously express these proteins outside of the cell. So one thing I'm not going to really go into much at all is genomic editing tools. We've tried quite unsuccessfully for a number of different ways, but this kind of work is ongoing, and we think there's been some promising movement on the CRISPR-Cas9 systems that could be used in Geobacillus. So the last portion of the talk, and just in these last couple of minutes, I just want to talk to you about some of the ways in the lab that we're trying to speed up this design, build, test, learn cycle. So obviously, the more iterations we can get through this, the more information we can get out, and hopefully the better our system will become. So one of the key things that we're using in the lab is lab optimization and miniaturization. So we recently purchased an OpenTrons robot, which Matt has been using quite extensively now to sort of play out some of his culture experiments, and hopefully we'll adapt this and use it a bit more within the lab. But yeah, trying to scale things down. We've done some experiments that compare plates with flasks. And we see some good correlation there, and also there's interesting systems that are coming online now, like the biolector system, where you can almost scale down these bioreactors to a world plate level. So it'd be interesting to see whether we can fold those into these and how well that translates. One of the things that I'm really keen on using in the lab is the design of experiments approach. So this came from my time at Exeter and Tom Howard over at Newcastle. But traditional experimentation takes a one factor at a time approach. So usually we have two things that we're looking at, or we're looking at a response to a change in enzyme concentration. We pick our highest level, and then we say, okay, well, what happens if I introduce another factor into that experiment? And I keep the original factor at that one level, and I change the rest, and I can see that I've managed to reach a new optimum. But I suppose what one factor at a time doesn't give you is the interaction between the two factors, and actually that's what design of experiment does. If I was able to look at those interactions, then we might be able to hit a new higher level. We're also trying to use this a lot more now for bioprocess optimization. So things where we're looking at not only the enzymes involved, but environmental factors. So you can see that we can quite easily get up to a huge number of experiments very quickly. So what DOE allows us to do is hopefully use a smaller subset and allows us to understand the interactions so we can hit these optimum a lot quicker. And we've actually tried to state this further now, where we look at some sort of different machine learning methods for dual optimization. And I'm helping supervise a PhD down at the University of Exeter with Oscar Ackman, where we're sort of trying to use these on a couple of test projects. And the last way we're trying to speed up this design cycle is by using cell-free systems. So obviously we don't have to worry about the timely transformation and bacterial growth process that can be quite time costly within the lab. There should be a reduced biological complexity. Obviously, we're not worried about the cell growing, so we should be able to see hopefully what are the direct consequences of our genetic circuits that we're putting in and things like that. And hopefully we should be able to scale these down, to the microfluidic scale, which will help us go through the greatest number of experiments. Obviously, cell-free systems, as we heard yesterday, have got much wider bioengineering projects that they're interested in. They're great for things like portable diagnostics, looking for nucleic acids or small molecules. And they're also now widely being used in biomolecular manufacturing. So things like the therapeutics, membrane proteins, or on-site reagents and things like that. So as you can see, it was a very introductory talk to some of the SynBio techniques and methods that we're trying to use in the lab. But hopefully as the hub grows and as we go through these summits, you'll start to see how we're using some of these tools and some of these organisms, and how hopefully we're starting to use some of this engineering biology to do some really interesting conversions of these waste products. So thanks very much.
Thank you, Paul. Right on time as well. Thanks very much for that. I've got one quick question. You talked briefly about insulator sequences. Could you expand on that a little?
Yeah. So I mean, there's some ribozymes that basically cleave the ribosome off between the ribosome binding site or after the ribosome binding site. So this is an idea that hopefully what it will stop doing is that direct read through, and hopefully what we'll start to see is the promoter having just the one level of effect on all coding sequences, and it not be so coding sequence dependent.
Okay, brilliant. Excellent. For the sake of timeliness, we'll move on to Shafir now, but thank you very much for that, Paul. Excellent.